Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 2, Bacterial Cell Structure and Function. In this extensive lecture, we will talk about bacterial cells, give you an overview of bacterial morphology, talk about the bacterial cell envelope, the plasma membrane, the concept of the cell wall, Gram staining, and further continue on talking about cytoplasm, endospores, mechanisms of transport, cell division, etc. And then we will end with a recap and conclusion. Now, bacteria are single-celled prokaryotic organisms, and they have unique features like they lack membrane-bound organelles. They possess specialized structures for survival and adaptation. Why is this important? Because understanding their structure helps explain their function, their pathogenicity, and antimicrobial targets. So developing antibiotics, for example. Now, overview of bacterial morphology. So let's talk about some shapes and arrangements. We have cocci. These are spherical. For example, the bacteria Staphylococcus aureus. And here is an illustration. We have bacilli. These are rod-shaped. You can see that here. For example, E. coli. We have spirilla. These are spiral-shaped, like Treponema pallidum, seen here. And Treponema pallidum, if you didn't know, is the one causing syphilis. Then we have vibrio. This is comma-shaped and causes vibrio cholera. It looks like that. Then we have pleomorphic bacteria, and these have a variability in shape, for example, mycoplasma. Now, cellular arrangements. So we can have chains, like for example, Streptococcus. These can be in clusters, like Staphylococcus, and they can be in pairs, like Diplococcus. So all in all, bacteria come in different shapes, sizes, and forms. Here then we have an illustration of these cellular arrangements. So as we mentioned, the chains seen in, for example, Streptococcus, we can see here the pairs in Diplococci. You can see it here. And of course, clusters in Staphylococcus, you can see illustrated here. Now let's continue on to bacterial cell envelopes. Now let's talk about the bacterial cell envelope. So what are the layers of this cell envelope? We have the plasma membrane, seen here. We have the actual cell wall, seen here. And then a capsule or a slime layer, if present. Now, what is the function of the cell envelope? Well, to protect, shape, and for selective permeability. So the cell envelope is like a bacteria's armor. It's protecting it from the environment. And as mentioned, it has multiple layers. And it has a membrane which controls for what goes in and out. And this envelope, what you need to remember is that this cell envelope is what makes bacteria tough and resilient. Now let's talk about the plasma membrane. So the plasma membrane is like the security gate of the bacterial cell. And it controls what nutrients and waste enter or leave. And it also helps the cell to generate energy. Now, some antibiotics work by damaging this gate, killing the bacteria. Now, its structure, it's a phospholipid bilayer with embedded proteins. Its functions, selective permeability, so transport of nutrients and waste. Furthermore, as we mentioned, the electron transport chain is here for energy production. And as we mentioned, why is this clinically relevant? Because it is the target of, for example, polymyxin antibiotics. Now, as we mentioned before, we said that the bacteria has this cell wall, and we differentiated between gram-positive and gram-negative, where gram-positive has a much thicker cell wall compared to gram-negative that has a much thinner cell wall. Now, the composition for the cell wall, whether it's positive or negative, is that it consists of peptidoglycan, also known as murin. And this peptidoglycan is a polymer made of two repeating sugar molecules that form a glycan backbone. Okay, so we will find sugar here, derivatives of glucose essentially. And this forms a glycan backbone. Now, these sugars then are linked to short peptide chains. All right? Short peptide chains. And it's these structures that make it both strong and flexible, allowing the cell wall to resist, for example, osmotic pressure while still being somewhat adaptable. So when we talk about peptidoglycan or murin, we have here the N-acetylglucosamine, NAG, and acetylmuramic acid, NAM. Now, NAG is a derivative of glucose. Indeed, the NAG is a derivative of glucose and is one of those repeating sugar units in the backbone of peptidoglycan, while NAM, or acetylmuramic acid, this is a modified form of glucose. And what differentiates it is that it has a lactic acid group attached to it. So it is the other sugar in the repeating backbone. Remember, we said that peptidoglycan, or murin, is a polymer made of two repeating sugar molecules, and these form the glycan backbone. So it's these two. And then we had these peptide chains cross-linking them. And here in the illustration, you can somewhat see. This is the peptidoglycan, right? Thick wall, because we're talking about gram-positive. All you need to remember right now is that it has a thick peptidoglycan layer. But you can see these cross-links here. Now, just to mention another thing, these peptide chains, right? These peptide chains are cross-linked to each other in this peptidoglycan mesh. And this cross-linking indeed is what provides additional strength and stability to the entire structure. Now, we have gram-positive. Indeed, it has a thick peptidoglycan layer. We also have teichoic and lipoteichoic acid here, as you can see illustrated here. While in gram-negative, we have a thin peptidoglycan layer. But we also have what differentiates it from the positive gram-positive bacteria is that it has an outer membrane with lipopolysaccharides, LPS. We will look at this in the next slide. Now let's put the gram-negative and gram-positive in comparison and dive into detail to differentiate between them. Hopefully, after this slide, you will have a list and a way to identify the differences between them. So we mentioned that starting with the cell wall structure, okay? Now, the gram-positive bacteria had a thicker or a thick peptidoglycan layer. You compare that to gram-negative, it is much thinner, as illustrated here. Furthermore, this peptidoglycan layer that is thick in gram-positive provides rigidity and strength to the cell wall. But another thing that differentiates the two is that gram-positive does not have an outer membrane. If you look at gram-negative, you find an outer membrane here. And this outer membrane, which is outside, right? It's outside the peptidoglycan layer that the gram-negative bacteria has, also then acts as an additional barrier. So that's one main difference, or two main differences. Then we have gram staining procedures, right? Where we want to identify when a cell is either gram-negative or gram-positive, or vice versa. And when we do a gram stain on these bacteria, if it's gram-positive, you will have purple or violet after gram staining. So it will turn purple or violet. And this is happening due to the thick peptidoglycan layer, which retains crystal violet stain. We use crystal violet as part of gram staining procedure. This is not something you need to learn or understand immediately now. We will briefly mention it as well in the next slide. But essentially, we apply crystal violet and iodine and alcohol and some other things, and the stain will then turn a certain color based on if it has a thick layer or a small layer. So it's worth repeating. This is high yield. This is asked in a lot of medical tests. So gram-positive, purple or violet. And why is it turning purple or violet? Because of the thick layer retaining this crystal violet that we are using. Now in gram-negative, it's going to turn either pink or reddish. And this is indeed due to the peptidoglycan layer. Because it's thin, it cannot retain the crystal violet stain. And instead, it's also part of the procedure, safranin, we call it the counter stain, that will color the bacteria instead, and it will turn pink or reddish. So pink or reddish, gram-negative, purple or violet, gram-positive. There we go. So that's two main things. The cell wall structure is different, right? One has an outer membrane, the other one doesn't. One has a thick peptidoglycan layer, the other one doesn't. One turns purple during staining, the other one doesn't. Third, we mentioned already briefly the outer membrane. So in gram-positive, this outer membrane is absent. In gram-negative, it's present. It's a double-layered outer membrane containing lipopolysaccharides. And this is contained on the outer leaflet, as you can see here. And it can actually trigger immune responses. So let's briefly check into this lipopolysaccharide that we have identified existing on the outer part of the outer membrane of the gram-negative bacteria. So lipopolysaccharides, first of all, are absent in the gram-positive bacteria, but in the gram-negative, they're present. And the lipopolysaccharide, particularly the lipid A portion of it, is a very potent endotoxin, and that can cause severe immune reactions in humans. And if you say, hey, it's unfair that the gram-negative has all of these things and the gram-positive doesn't, well, the gram-positive has something else. It has these teichoic acids, and these are embedded inside of this thick peptidoglycan layer. And these acids help with the cell wall maintenance and contribute to the negative charge of the cell. And the gram-negative don't have that. Instead, they have these lipoproteins in the outer membrane. Another difference is the periplasmic space, seen here, right? And in gram-positive, it's very narrow or even absent. And there is only a small space between the plasma membrane and the thick peptidoglycan layer, as you can see illustrated here. This layer, this space here is very, 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 very small. While in gram-negative, right, in gram-negative bacteria, it is present, and the periplasmic space is very well defined and located between the inner membrane and the outer membrane. And it contains enzymes and transport proteins, etc. Now let's turn a bit more to the clinical part of all of it. So what is the difference in sensitivity, for example, to antibiotics? So gram-positive bacteria are more sensitive to antibiotics like penicillin. Why? Because penicillin targets peptidoglycan synthesis. So it is more easily targeted because their thick peptidoglycan layer is more exposed, right? It's bigger, it's more exposed. And gram-negative is less sensitive to many antibiotics due to the outer membrane that it has, right? It has an extra outer membrane here acting as a barrier, so it makes it harder for antibiotics to penetrate and reach this peptidoglycan to cause its harm, or in another way, to heal us. What about resistance to phagocytosis? Well, gram-positive can be susceptible to phagocytosis, although the thick peptidoglycan layer sometimes make it harder for immune cells to actually engulf it. And the gram-negative ones, especially the lipopolysaccharides that are here expressed on the outer surface of the outer membrane, they can act as a shield against our immune system, so it makes them more resistant to phagocytosis. And if you don't remember phagocytosis, essentially you have these cells, macrophages, scouting around for pathogens or something that should not be in our body. And if it detects it, it will try to eat it up, engulf it, and break it down inside and then spit it out. So that's the context here. Now, let's see an example here. We have Staphylococcus aureus or Streptococcus pyogenes or Bacillus anthrax. These three are examples of gram-positive bacteria. Some gram-negatives could be E. coli or Salmonella or Pseudomonas aeruginosa. That's a gram-negative. And you have probably heard of these names before. You've heard, oh, somebody has caught Salmonella, or the person has been diagnosed with E. coli or Staphylococcus or whatever. Now you will always remember that, oh, these actual bacteria that we have names for are actually differentiated to being either gram-negative or gram-positive. And if they are put into these different categories, I can't approach them clinically the same way. So it's not, oh, this is a bacteria and this is a bacteria, give them antibiotics and it's resolved. It's more sophisticated. It's more which type of antibiotic and why. What are we targeting? And to make sense of this, you have to understand first that these are categorized into different groups. And then these groups have different structures. And due to these structures, they will adapt and behave and respond differently to whatever drug we give them, etc., etc. Now, let's indeed mention gram staining. So gram staining is a procedure where we apply crystal violet, which is the primary stain. We then add iodine to fix this stain. Use alcohol to wash it and decolorize it all. We then add safranin, which is the counter stain. When we have done all of that, we look at the results. Again, gram-positive turns purple or violetish. And that's because it retains crystal violet. And this is the high-yield question. Why is it retaining it? Or how is it retaining it? Exactly, because of the thick peptidoglycan layer. And then we turn around and say, what about gram-negative? Why is it turning pink or reddish? Because it gets stained by the counter stain, safranin. So how is the safranin beating the crystal violet? Because, exactly, the peptidoglycan layer in the gram-negative is much thinner. So it cannot retain this crystal violet, and instead safranin takes over. Now, we already touched upon the outer membrane and the lipopolysaccharides. And if you remember now, we are talking about gram-negative bacteria, right? It has this outer membrane, first of all, that the gram-positive does not have. And expressed on its outer layer, it had these lipopolysaccharides. And you can see here an illustration again. This is the outer membrane, right? And this is the LPS expressed on it. Now, the structure is lipid A, which, if you remember, was an endotoxin, causes inflammation and fever. It has a core polysaccharide, an O antigen, so it has antigenic variability. And the function of this membrane and LPS is to protect against bile salts, against antibiotics, against our immune system. Remember, it can be resistant to phagocytosis. Now, the clinical relevance here is that LPS is a virulence factor in E. coli and, for example, Salmonella. So all in all, gram-negative bacteria have an extra outer layer acting like a shield, so it protects it, making it harder to destroy and kill. But also, it contains LPS, lipopolysaccharides, which is a molecule that causes severe inflammation or can cause severe inflammation and fever in humans when these bacteria infect us. Now, capsules and slime layers. So capsules are like invisible cloaks, right, for these bacteria. It helps them to hide from our immune system. And it makes the infections harder to treat. Now, what are some characteristics of this capsule? Well, it is a thick, organized polysaccharide layer. Its role is to prevent phagocytosis, and it helps in adhesion. An example of this is Streptococci pneumoniae capsule, which enhances virulence. What is the slime layer? It is a thin, unorganized layer aiding in biofilm formation. So, essentially, slime layers are sticky coatings, right? Sticky coatings that help bacteria to stick to surfaces and form a protective communities or something. So biofilms. And the clinical relevance here is that biofilms are resistant to antibiotics. Appendages. So bacteria have tiny structures called flagella or tails, and these are used to swim. Or they have something called pili that are spikes, and this helps them to stick to surfaces. And some pili are even used to transfer DNA between bacteria, helping them sharing tricks like antibiotic resistance, for example. So the flagella structure is of a filament, has a hook and a basal body. Function, indeed, motility and chemotaxis. An example is Helicobacter pylori, which uses flagella to colonize the stomach. We have again then pili. Its role is adhesion to surfaces, conjugation, so transfer of plasmids. Example could be Neisseria gonorrhea, which uses pili to adhere to host tissues. Now, what about the cytoplasm of the bacteria? Well, it's composed of the cytosol, ribosomes, nucleoids, inclusion bodies. Now, this is super important. The ribosomes in prokaryotic cells are 70S versus eukaryotic, it's 80S. Now, what does this mean? So first of all, we're talking about ribosomes, and these are essential cellular structures responsible for what? Protein synthesis through the process of translation. You will learn about this in our course on cell biology, but it's worth repeating. So they're important for protein synthesis through translation. And this is where the genetic code from mRNA, messenger RNA, is used to build proteins. Now, these ribosomes, connecting this to this now, these ribosomes consist of two subunits. And we have a large and a small subunit, which together form the ribosome's overall structure. And the ribosome's size, okay, its size is measured in something called Svedberg units. And the unit used is indeed S. So this is 70 Svedberg unit, and this is 80. That's number one. And the Svedberg units indicate the rate of sedimentation during a process called ultracentrifugation. And roughly, they also correlate to the size and the shape. So 70S and 80S refer to the sedimentation rates of the ribosomes in their respective organisms. So the prokaryotic ribosome, 70S, if we start with that, it is composed of two subunits. It has a small subunit called 30S, and it has a large subunit called 50S. And these are composed of different amounts of RNA and proteins, etc. We don't need to go to too much detail here, but the structure is essentially that this 30S subunit, okay, of this prokaryotic is what binds with the messenger RNA to read the genetic code, right? And the 50S subunit is where transport RNA molecules bind and amino acids are linked together to form the growing protein chain. So, one example would be E. coli. So, this is very directly important and relevant to the whole composition and structure of the bacteria. These ribosomes responsible for protein synthesis in prokaryotic cells, so bacteria, we have 70 Svedberg units. It is then divided into 30S and 50S, and these mean something. They're also very relevant later for treatment processes, but here we know then that 30S was what binds to the messenger RNA to read genetic code, and 50S is where the molecule bind and amino acids are linked together to form the growing protein chain. Now, what about the eukaryotic cells? Here we have 80 Svedberg units, right? Now, 80S ribosomes in eukaryotes are larger than prokaryotes, and they consist of their small subunit is 40S, and its large subunit is actually 60S. And similar here to the prokaryotes, the 40S subunit is what binds to the messenger RNA to read the code, and the 60 subunit forms the site for protein elongation. And the eukaryotes is what we find in humans. Prokaryotic is in bacteria. Now, why does this matter? Why did we jump into the structure of the bacteria, focusing on ribosomes, differentiating between their Svedberg units, finding their subunits, differentiating between 30 and 50S versus 40 and 60? Why does this matter to us as medical doctors? Well, listen, the difference in ribosome size between prokaryotes and eukaryotes is medically significant because antibiotics have evolved to target bacterial 70S ribosomes without affecting the larger 80S ribosomes in human cells. So there has been discoveries whereby we are able to attack this group without attacking this group. So this is key for their effectiveness for antibiotics in treating infections without harming the patient's own cells. So let's just talk briefly about some examples. We have an extensive lecture on antibiotics, but now just to give you an overview. So we have a class of antibiotics called aminoglycosides, and these contain, for example, gentamicin, streptomycin, maybe you've heard of them. Now, these aminoglycosides, they bind to the 30S subunit, preventing the attachment of the amino acyl tRNA, thereby inhibiting protein elongation. Excuse me, that's not right. What they do is they bind to the 30S subunit of the bacterial ribosome, causing misreading. That's what they do. They cause misreading of the mRNA inhibiting protein synthesis. If you do like I did, which is for a moment they're confused what these two did, you will then think that the antibiotic we use targets the wrong one. But we had to correct ourselves, right? And say that no, we remember that 30S subunit is what is involved in reading the messenger RNA for inhibiting protein synthesis. And the 50S was involved in the process of the leading to the elongation of the protein chain. So the aminoglycoside group, like streptomycin, will bind to 30S, causing a misreading of this messenger RNA and therefore leading to an inhibition of the protein synthesis. So it can't produce more, right? Another group of antibiotics are tetracyclines. For example, doxycycline. Now, what they do is they also bind to the 30S subunit. But what they do is they prevent the attachment of this amino acid tRNA, thereby indeed inhibiting protein elongation in the bacteria. Another group we could mention is macrolides or macrolides, like erythromycin. What do? They bind to 50S subunit of the bacterial ribosomes, preventing formation of peptide bonds during protein synthesis. No, actually scratch that. We just committed the same error again. The macrolides bind to 50S, yeah, that's correct, but they prevent them from, they are preventing the elongation of the protein chain. The other group is chloramphenicol. It inhibits 50S subunit of bacterial ribosomes, preventing the formation of peptide bonds, indeed, during protein synthesis. So to summarize then, we have 70S ribosomes. Actually, let's clear this up and say we have 70 Svedberg unit ribosomes found in prokaryotic cells, so bacteria. And these are, number one, smaller than eukaryotic. And they are composed of 30S and 50S. Now, don't be surprised when you encounter this kind of question in your medical exams, because this is indeed questions that will resurface many times through your medical studies. So it's worth mentioning here and repeat. Furthermore, why is it different from eukaryotic? Because eukaryotic had its small subunit of 40S and 60S. This can sometimes be a bit confusing because you see 80 and you think that this is the sum of these two, but it's not. And 70S, I guess 70 is not a sum of 30 and 50. This is just one of those things where you just have to go with it. And another high yield thing was that this smaller subunit was involved in the messenger RNA process, essentially binding to the messenger RNA to read genetic code. And the large subunit is where the tRNA molecules bind and amino acids are linked together to form the growing protein chain. And it's similar in prokaryotic as in eukaryotic. We also learned that antibiotics are developed to target these different subunits, leading to prevention of different processes. And the whole purpose of all of this and knowing all of these details medically is that this difference between them, between the prokaryotic and the eukaryotic, is leveraged to develop antibiotics to selectively target bacterial ribosomes and minimizing damage to human cells, ensuring the effectiveness of treatments. So that's that. Another thing worth mentioning then is the nucleoid, which is also here present. You can look at this illustration here in the cytoplasm, and it contains the bacterial chromosome, which is single and has circular DNA structure. Why is it clinically relevant? Well, plasmids carry antibiotic-resistant genes. So that's something we need to keep in mind as we continue developing antibiotics as ways to get around it. Now, what are endospores? These are dormant, highly resistant structures formed under stress. Some examples can be anthrax or botulism, Clostridium botulinum. And some bacteria can hibernate, indeed by forming these tough shells called endospores. And like we said, dormant, highly resistant. So these spores can survive extreme heat or dryness or chemicals, etc., making them hard to kill. And what they do is, very sneaky, they are waiting until conditions improve to then get out and become active again. So this resistance is part of that thing. Now, why is this clinically relevant? Because indeed, they are difficult to eradicate. So we require high-level disinfection to deal with it. Now then, we mentioned in the beginning that we will talk briefly about the mechanisms of transport, because bacteria also needs to bring in food and get rid of waste, and they use these different gates in their membrane to move things in and out. And some bacteria even use these gates to actually pump out antibiotics, making them resistant to treatment. Now, we're just going to briefly mention a couple of mechanisms. So we have different types. We have passive transport and active transport mainly. In passive transport, what is important to remember is that energy is not required for the utilization of these transport systems, while in active transport, energy is used. So in passive transport, molecules move, right? Molecules move across the bacterial membrane without using energy, without using ATP. And it's driven mainly by concentration gradients, for example, from high concentration to low concentration. And it's important for bacteria because it allows it to efficiently take up small molecules like oxygen and CO2 without expending energy. And this is critical for survival. Then we have active transport. Now, in active transport, the bacteria move substances against their concentration gradient. So from here out, let's say. And this requires energy, usually in the form of ATP. And this is essential for taking up nutrients like sugars, amino acids, and ions in environments where these are present at lower concentrations than inside the cell. It also allows bacteria to expel harmful substances like antibiotics or waste products. This contributes to antibiotic-resistant mechanisms. We have another one we didn't mention here, but it's called group translocation, and it is a unique transport mechanism where the substance is chemically modified during its transport across the membrane, and it allows the bacterium to trap essential nutrients like glucose inside the cell in a modified form that can't easily leave, ensuring the bacterium can use these for their metabolism. So efficient transport is medically incredibly relevant because it influences how bacteria obtain nutrients, expel toxins, survive in hostile environments, including in the presence of antibiotics. Now, what about bacterial cell division? So the process of bacterial cell division is binary fusion. And we have these steps here. And you should try and remember these. So first, we have DNA replication, right? And the chromosomal segregation is happening during the same time. Now, after that, we have septum formation. And finally, we have cell separation. And The time required for a bacterial population to double is called generation time. For example, E. coli divides every 20 minutes under optimal conditions. So to summarize, bacteria reproduce by splitting into two, like copying and pasting themselves. The process is called binary fusion, and it's incredibly fast. Like some bacteria, for example, E. coli, can double every 20 minutes. That's why infections can grow very quickly if we don't treat them. Now, clinical relevance then. We've already touched upon a lot of these things before, but let's repeat. So understanding bacterial structure informs us about antibiotic targets. Peptoglycan, are we targeting the layers? For synthesis, are we targeting the ribosomes? Subunit, which one, etc. We need to be aware of the mechanisms of resistance, like the bacteria using efflux pumps, the lipopolysaccharide, essentially whatever it is repelling our immune system from destroying the bacteria, or when we give it antibiotics, what throws it out of the bacteria. These pumps, for example. We have mentioned the importance of diagnostic techniques to be able to identify, is it a gram-positive, is it a gram-negative? And if you remember, we used crystal violet. And if crystal violet remained due to the thick layer, it indicated then a gram-positive. And some clinically relevant drugs would be beta-lactams, like penicillin. They inhibit peptidoglycan synthesis. We had another group called aminoglycosides. These were targeting bacterial ribosomes. So in summary then, bacterial cell structures are diverse and specialized for survival. Each structure contributes to pathogenicity, survival, or antibiotic resistance. Understanding these structures help us design diagnostic tools and better therapeutics. And that's the end of this lecture. Hope it was interesting. Continue now to Chapter 3.